Yeah. No problem. So we've got DNA here. We know DNA is a nucleic acid. What are some other nucleic acids? ATP, GTP, yeah, like NAD plus, NADH, FAD, FADH2, all those electron carrier things. That's a really nice look for you, by the way, Maddie. DNA, like all nucleic acids, is made of building blocks called nucleotides. And remember that a nucleotide is made of 3 pieces itself. A nucleotide is made of, in the middle, what's that little green ring thing? A sugar. And in DNA, that sugar is deoxyribose. Attached up to one side of that sugar is the phosphate group, and attached to the other side is the nitrogenous base. Now, remember the numbers up here. What do they number? The carbons in the sugar. So the phosphate's always attached to carbon number 5, and the sugar, or the nitrogenous base rather, is always attached to carbon number 1. Also, while we're looking at the strands of DNA, remember that they're sort of opposites of each other. Notice this one, the point is pointing up. This one, the point is pointing down. This one with the flat side at the bottom is called the 3' end. Wherever the pointy side's at, that's where the 5 carbon's at. So up here, this will be the 5'. It's opposite over here. Points down here, so that's 5'. So this is 3. And since they're kind of parallel but opposites, we say they're what? Anti-parallel.
Is Katie Schwartz in the building? Please go to Mrs. Fanny's office.
Also remember that what holds these 2 chains together is that the nitrogenous bases are complementary. A goes with T, G goes with C. And the little dots represent what? Hydrogen bonds. Remember that A and T only form 2. G and C form 3. So A's and T's are easier to break apart than G's and C's are. There's less bonds. All right, I'm going to skip the history stuff for right now, I think. All right, the key things to know about DNA. It's 2 chains of nucleotides. They're anti-parallel to each other. The backbone of DNA is made out of alternating phosphates and sugars. It's just phosphate, sugar, phosphate, sugar, phosphate, sugar. Can that be the important part? It's just the same thing over and over again. Can't be, right? It's the same in every living thing. Bacteria, virus. The virus is not even really living, but it's the same there too. Plant, animal, fungus, and you. It's the same in everything. The important part of those bases. And those bases make up a code. And what do they code for? Proteins. What is the smallest unit of the code called? A codon. And it's made of how many base pairs? 3 base pairs. Each codon codes for what? 1 amino acid. So again, here we see the backbone of the DNA. But here's the important part. Let's focus, please, guys. Remember that DNA's only real function is that when they're in bulk. Codes for proteins. And then what do proteins determine about you?
Everything.
Everything. And what are your body's most important proteins? Your enzymes. Catalysts that make your body work. Go a little further there. Differences between bacterial, prokaryotic, and eukaryotic DNA. Prokaryotic chromosomes are circular. They're really small. There's usually only 1 of them in every cell. And they may have some enzymes and proteins attached to the DNA, but not very many. And they're just free floating out in the cytoplasm. What's the DNA of a prokaryote sometimes called? Nucleoid, which really means without what? Without a nucleus. Whereas eukaryotic DNA is said to be linear. Our chromosomes are linear. They're not really straight lines, but they're not loose. They're way longer than the bacterial ones. There's usually several per cell. In us, how many per cell? 46. Our DNA is attached up to these proteins called histones, which act like spools that our DNA winds around. Eukaryotes have histones. What other group has histones? Archaea. That's one of the reasons scientists think we're more related to them than we are to the regular OU bacteria. Our DNA is enclosed in a nuclear envelope, double membrane nuclear envelope. One thing I'll mention about the histones. If our body needs to turn on a gene, what does it do to the histones sometimes? It adds an acetyl group, a 2-carbon group to the histone. And what does that cause to happen to the DNA around that histone? It unwinds, and therefore what 2 things can happen to it? Transcription and translation. That means it's on. Now if our body wants to permanently turn off a gene, what do we do to the DNA? Add methyl groups to it. And that causes that DNA to do what? Wind up in tight knots that don't ever come undone. And that causes cells to specialize, where you turn off certain genes and you don't turn off others. Going a little further, the whole central dogma of genetics idea, that at its most basic part, we start with DNA. With that DNA, what do we call the DNA? What kind of blueprint? $1 million blueprint. And $1 million blueprint is not something you take out and show off. You keep it locked up somewhere, right? And in us, that DNA is locked up where? In the nucleus. It can't get out. But the protein-making factories, which are what? The ribosomes, they're not in the nucleus. So we've got to get the directions from the nucleus to the ribosomes, and we do that through what? mRNA. And we know the process of using the DNA as a template for making mRNA. That's called transcription. Now that mRNA will leave the nucleus, and it'll go out to the ribosomes. How does it get out of the nucleus? It's got pores, holes. And it'll start getting read and decoded, and it'll be used to build the proteins. That process, mRNA to proteins, that's called what? Translation. Now remember, you got 2 different kinds of ribosomes. You got ones that are attached to the ER. If the mRNA goes there, what's going to happen to the proteins that you make? They're going to get shipped out, and that's called what? Exocytosis. And the system that makes it and ships it out, that's called what? Endomembrane. If it's made on a ribosome that's just sort of floating around in the cell, where's that protein going to end up? It's going to stay in that cell and do something inside the cell that needs it. Once we make our proteins, we know that they determine our traits. Everything about us. And then the one thing that we're leaving off here, if we put a little loop here, that would represent what? Replication. And when in a cell's life does the DNA need to replicate? In this phase of interphase. Just a little bit before what starts? Mitosis, or maybe meiosis. Now, as far as we know, every living thing does this. That's why it's called the dogma. It's a main idea. But there's one kind of thing that's not really living that's an exception to this rule. And what is it? The retroviruses. Because they only have what kind of genetic information? RNA. So they inject RNA into their host cell, whatever cell they're trying to invade. And what do they inject along with that RNA? Yeah, an enzyme called reverse transcriptase. And that enzyme will take the RNA and use it to make DNA. So it's backwards. So they have an extra step. And what do we call those viruses? Retroviruses, or RNA viruses. And what are some examples of those? Common cold, Ebola, influenza, Marburg, There's probably some others, but those are the main ones. What we're going to talk about here now is replication. We know again, replication is when the DNA is copied. A key thing to know about replication, first of all, when it happens, at a phase of interface. Also, you need to know that DNA replication is semi-conservative, meaning that when the DNA strand unwinds, each old strand acts as a what? A template. And then we end up with two new double-stranded pieces. But each of these two new double-stranded pieces consist of one what? One old strand and one new strand. And that helps to make sure that what doesn't happen? Mutations. Because that old strand acts like a what? Template. And that's what's called semi-conservative. Conserve means to stay the same, not to mutate. And the semi means that each new double-stranded piece is half what? Half old, half new. And that helps to prevent the mutations. But some key things to know about replication. What's the first enzyme that sort of kicks it off? Helicase. Helicase does what? Unzips, unwinds the DNA. And what do we call the place where this starts? Replication fork, or origin of replication. And on each eukaryotic chromosome, how many of those are there? Lots. But on a bacterial chromosome, there's only what? Only one. Helicase unwinds the DNA. Step 2 is an enzyme called primase has to come in and put a little piece of RNA on there. Sort of a start here kind of thing. What's the main enzyme involved in replication? DNA polymerase. It actually builds the DNA from little nucleosides, little spare parts out in the nucleus. But it can only put them in what order? 5 to 3. So this strand, this dark purple strand here, that's the original strand. It's running from 3 to 5. But the strand we're going to build onto that has to be 5 to 3. That's the way DNA likes to work. DNA polymerase, that's 5 to 3. So this strand gets put together in one continuous piece, non-stop, and therefore it's called the leading strand. But this other strand, this original strand starts with 5 and goes toward 3. The strand that we're building is going to have to run from 3 to 5. Polymerase can't do that. So it has to skip forward and work back this way, 5 to 3. And then it skips forward here, works backward, 5 to 3. And it does that in pieces, chunks. And what do we call those chunks?
Okazaki fragments.
Okazaki fragments. And right now they're not connected. Eventually, what enzyme is going to put all those fragments together into one piece? Ligase. And when we get finished, this piece up top will be a double-stranded piece that will be exactly like this piece down here. That's why we call it semi-conservative. But it takes, the strand on the bottom is a little harder to make because we have to do it in discontinuous pieces of Okazaki fragments. Does that make sense? There we see replication happening in a eukaryote. It starts at multiple places, multiple origins of replication. Here we see it in a bacteria. It starts at only one place. Let's talk about RNA for a minute. Single-stranded. Instead of having thymine, it instead has uracil, but it's still a nucleic acid. It's still made out of nucleotides. Nucleotide here, 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 here. There's several kinds. What's the one we talk the most about? The kind of RNA that we talk most about. mRNA. And the M stands for? Messenger. Because it's a messenger between what? And what? Nucleus and ribosomes. DNA and ribosomes. We also talk about tRNA. What's the T stand for? Transfer, but I told you to think of it like what? Yeah, a taxi. Because its job is to deliver what? Its job is to deliver the amino acids to the right place during protein synthesis. It's got to be really, really specific about that. And then the other kind of RNA that we'll briefly mention is rRNA. What's the R stand for? Ribosomal. Because that's what ribosomes are actually made from. And by the way, where are ribosomes made in a cell? They're made in the nucleolus. Yes. Hey. Again. So, ribosomes are made in the nucleolus, and then they move out, and they either go to an ER or they just float around in the cell. One of the two. But what we want to talk about now is transcription. Transcription is the process of using DNA as a template for making, before that, RNA. Transcription is the making of RNA. Now, when we're talking about DNA being used to make RNA, is one or both strands of the DNA used? Just one. What's the strand that we use to make the RNA called? The template. Now, key thing to know about that template strand is that the template strand is the one that has to run which way? The template strand of DNA is the DNA strand that goes 3 to 5. Because just like DNA polymerase, RNA polymerase can only build RNA which way? 5 to 3. So the strand it's building from has to be 3 to 5, so that the RNA it's making can run from 5 to 3. We'll work an example of this in a minute, make sure you know how to do it. But here we see that only this strand, only the template strand, is being used to make the RNA. When it's finished, what's going to happen to the 2 DNA strands? Once transcription's finished, what's going to happen to the DNA strands? They're just going to sit back together. Okay? And the RNA will go out and get translated. All right, let's go ahead and talk about a couple of terms here. There's 3 steps involved in transcription. The first one is initiation. How does RNA polymerase know where to start? What? Well, it depends. But there's this part of the DNA in front of most genes called the Tata box. There's a lot of T's and A's in there, T, A, T, A, T, A, so on. And that basically says start here. Begin the gene here. And that's where it starts. That's called initiation. And then we get what's step 2? Elongation. And that's when they add in the nucleotides. They add in the A's and the U's and the G's and the C's. And then what's last? Termination. And that's when the RNA polymerase gets to a code that says what? Stop. You're done. Once the RNA is made, then it's time to translate it, to do protein synthesis, to actually build it. That mRNA will leave the nucleus, and again, how does it get out? Through the pores, and it goes to the ribosomes. And the ribosomes will attach, again, in a kind of a 3-step process. First, we get chain initiation, where the RNA attaches up to the ribosome. Remember, the ribosome's in 2 pieces. Focus, guys. Large and small. How does it know to start here? Remember, the RNA has a cap on it. Let's back up for a minute. Between transcription and translation, what has to happen to the RNA? It has to get processed, which means what has to get removed? The intron sequences, the non-coding sequences. What are the enzymes that take those out called? Spliceosomes or SNRPs. And also during that process of processing, a cap gets put on the 5' end that basically says start here. And what gets put on the other end? Tail, made of A's, right? Of adenines that basically says don't destroy this. It's good. So when we get ready to do translation or protein synthesis, the ribosome attaches up to the mRNA just behind the cap, the 5' cap. The first codon on the mRNA is always AUG. That's the signal to start. tRNA will bring in the very first amino acid. It turns out it's always methionine. Because the only thing that can park in this spot right here is a tRNA with what? A UAC. What are those codes called on the tRNAs? Anticodons. And they have to match up with the codons on the mRNA. If they don't match, it won't work. A tRNA with this codon is always going to be carrying what? Methionine, every time. Guys, we got to focus here. The next thing that happens is that the big part of the ribosome comes in and binds. And then after that, the next tRNA is going to come in. What anticodon does it have to have? CAU, right? No T's in RNA, right? No T's. So it's got to be CAU, and that's going to bind there. And that's step 2 of translation, which is what? We initiated, and now we've got to elongate. Whoops, got ahead of myself there. And elongation in this case means we're elongating what? The amino acid chain. The next tRNA comes in here, and it brings in valine. And that bound up to the methionine right there. What's that bind between amino acids called? Peptide bind. And it forms between what 2 functional groups?
Carboxyl, hydroxyl.
Yep. A carboxyl on the left one to an amino on the right one, right? Carboxyl over on the left to amino over on the right. What's going to happen with this tRNA that just left? It's going to go reload. And where does it get its amino acids from? From your food, mostly. And that process is just going to continue to happen. New tRNA is going to come in, bring in amino acid. They get bonded to each other, peptide bonded to each other. Then the tRNA will leave and it'll go reload. And that's going to continue to happen until you get to a code that eventually says what? Stop. And that's termination. And when it hits termination, everything comes apart. And this chain of amino acids that we have now is now called what? Polypeptide. And if several of those get all wrapped together, then we've got what? Then we've got a protein. 2 other things to say about mRNA. Is it only used once? The mRNA. It's reusable, and it can actually be used by what? Multiple ribosomes at what? The same time. And what's that called? It's called a polyribosome or polyribosomal RNA. Lots of ribosomes working on the same piece of mRNA at the same time. All right, we're going to work through the sample problem. Before we do, though, if we're talking about mRNA, 3 nucleotide sequences on an mRNA are called codons. On a tRNA, they're called anticodons. And the table that we're going to use to help us do this, this table, is called what? The genetic code table. And the genetic code table is based on which sequence? mRNA. Unless they tell you differently, it's mRNA. And it'll let you figure out the amino acid sequences. So let's go to this problem at the end here. So we're given the DNA template sequence. And we know it's a template, number 1, because it tells us it's a template. But number 2, because it starts with what? 3. Starts with 3. The first thing I'm asking you for here is the non-template sequence. If the template sequence is 3, the non-template sequence has to start with 5, because those 2 strands are in parallel. The first codon is TAC, so the first anticodon has to be ATG. We're talking about DNA, no U's. All right, then we've got GGC, so our next codon has to be CCG. And then we've got CAA, so we need a G and a TT. And then we've got ATT, so we need TAA. And we're going to end with what? 3 prime. All right, now this is where people get mixed up. Okay, let's grab the mRNA. It is built from which one of these?
The mRNA.
The mRNA is built from which one of these? It's built from the template, right? So when you're trying to figure out your mRNA, forget about this sequence right here. You've got to think about that sequence right there. The template always starts with 3, which means the mRNA is always going to start with 5. If the first sequence on your template is TAC, the first sequence on your mRNA has to be AUG. The next set is GGC, so this has to be CCG. The next one is CAA, so we need a G and a what? UU. Remember, no T's in RNA. And then we get to ATT, so we need UAA. And then we end with what? 3 prime. All right, then we get down here to the tRNA. Now, one thing to remember about tRNA is the tRNA is in one sequence, is in one piece. You know, each separate tRNA molecule only has how many nucleotides on it? Each separate, let me back up and show you a picture. Yeah, let me show you a picture here. That's what tRNA looks like. It sort of looks like a cross. On one side of the cross is the anticodon. On the other side is the amino acid. The question I was asking is that the tRNA only has 3 bases on it, right? Each tRNA is going to be a separate thing. It's not one strand. So when we're doing the number thing on this, is there any need to put 3s and 5s on the tRNA? It's separate pieces. There's really no need to do that. So if we've got AUG as a codon, what's our first anticodon have to be? UAC, right? They have to match up. They have to be complementary. And if our next codon is CCG, that's to be G, G, and C, right? And then if we've got GUU, we got to have C and what? A and A. And then if we've got UAA, we have to have A and what? UU. So those are our anticodons found on tRNA. And then the final thing I'm asking you to do on this is to find the amino acid sequence. And to do that, we've got to go back to the table, right? So I'm going to write the sequence of the mRNA, which is what we're going to base that on, right over here on the board. AUG, CCG, GUU, and UAA. And we're going to use that mRNA sequence with the table. So let's back up to the table. And there we go. So we look up AUG. First base A, second base U, third base G, puts us right here. Methionine. So our first amino acid is methionine. There would be a carboxyl group sitting right here, right? And there would be what sitting right over here? Amino group. All right, we look up our next codon. CCG. CCG, right there. Proline. Over on the left side of that proline would be what group? Amino. And over on the right side, carboxyl. What kind of reaction is going to happen between the carboxyl on the methionine and the amino on the proline? Well, what? Okay, synthesis, give me some other names. Dehydration, give me one more. Condensation. And what's going to happen is this OH from the carboxyl is going to come off, and one of these H's are going to come off. And that frees up a bind site on both, and they bind. And we call that bind a peptide bind. It's a covalent bond, strong, hard to break. Then we read GUU. GUU is valine. And again, on the valine, on the left side, we would have amino. And on the right side, carboxyl. And the carboxyl on the proline and the amino on the valine go through a condensation or synthesis reaction. They form a peptide bond. Our last codon is UAA, and that says what? Time to stop, time to terminate. So everything will break apart, and this chain of methionine, proline, and valine, that is now a what? A polypeptide. And several of those are going to get joined and twisted and wrapped together to form the tertiary structure of what? A protein. That makes sense? Oops, didn't want to do that just yet. All right, so let me look at one other thing here. Remember that when you're doing transcription, only one strand is used to make RNA. And what again do we call that strand? The template. Which means in terms of numbers, if this top strand is the template, what's this number right here have to be? 3. And this has to be 5. Which means this strand right here, this has to be 5. And that has to be 3. What's the main enzyme that does transcription? Try again. RNA polymerase. And it only can build RNA, which way? 5 to 3. Both enzymes can only build 5 to 3. You're doing better today. Doing better today. All right, that's a good place to stop. Through a little early today.